You. I welcome you all to this Biotechnica's YouTube channel. I hope every one of you is fine there and you are preparing well for your QA exam. So, all right. So, everyone, please type yes if you can hear me and see me. Yes, cute princess, very good evening. Please type yes, all of you, if you can hear me and see me. Yes, Sneha, very good evening. Welcome back, Sneha. So today we are going to learn about recombinant DNA technology as well as genetic engineering. So today we are going to understand what is genetic engineering, what are the methods that we can engineer our genetic material, that is DNA, and what are the processes behind it. And the processes that we are using to modify this or alter this DNA, it was is what called as recombinant DNA technology. So firstly, we'll understand this genetic engineering and then we'll go to recombinant DNA technology. So, firstly, what is genetic engineering? So, the name itself suggests engineering. That means we are modifying or altering something. What we are altering? We are altering this genetic material. And the genetic material that we are going to engineer is DNA or RNA. So, can we say this genetic engineering is a technology that is used to alter this genetic material? Yes. So the technique in which genetic material, that is DNA and RNA, it is chemically altered, it was what called as genetic engineering. So how can we alter this DNA? How can we alter? We can alter this DNA with the help of chemical agent. And the agent that we are using is restriction enzyme. The structure you can see in the diagram, it is a restriction enzyme. So what all you have to do for the genetic engineering? So you all have studied about the tools of genetic engineering. What all is needed? We need restriction enzyme, that is RE. Different types of restriction enzymes are there, and the most common restriction enzyme that we are going to modify or cut this DNA, it's what called as RE2, that is restriction enzyme second. So what is done here is we will use this restriction enzyme to cleave our foreign DNA. And once it is cleaved, now we are having this foreign gene or gene of interest. We will clone it in a vector. So you can see a round circle diagram. This is a plasmid. So here plasmid is used as a vector or a carrier. So this will take this foreign DNA and now we are having a recombinant DNA, a gene of interest that is cloned in a vector. So that's why it is mentioned here, it is chemically altered, because here we are using restriction digestion or restriction enzyme second to digest our gene of interest. That will be introduced into the host organism, that is it can be insect, it can be E. coli, it can be anything that will change the phenotype. Why change the phenotype? Because we are using particular gene of interest and thereby the phenotype or the physical appearance will be changed because this gene will encode for some other proteins. So now we will look for the processes of RDT, that is recombinant DNA technology, or in short, we can also say it as a genetic engineering. So what are the processes that are needed for RDT? So the very first process is isolation of genetic material. Firstly, you have to isolate your genetic material. This genetic material, it can be in the form of DNA or it can be in the form of RNA. So firstly, we have to isolate this genetic material. And once this genetic material is isolated, again, it is needed that we should do the restriction digestion of this DNA. So what we'll do, we'll incubate this DNA with the help of a restriction enzyme, that is RE2. Any restriction enzyme we can do or we can use, it depends on our experiment. So cutting of DNA with the help of restriction enzyme at specific locations. Why specific locations? Because we know that it is called as restriction enzyme. Restriction enzyme, an enzyme that will cut over the restricted part. That is why it is called as restriction enzyme, an enzyme that will recognize a specific sequence. That is why it is called as restriction enzyme. And what sequences it recognizes? It recognizes palindromic sequences. Now, what is palindromic sequences? You have already read in the tools of biotechnology. This is what called as the sequence that will read the same forward and backward. Suppose I'm writing the Malayalam, Malayam. So if you read this word Malayalam, it will read the same forward and backward. So we have to use this kind of sequences for restriction digestion. So it will recognize the specific location. Now, once we are getting this gene of interest, what we'll do, we'll amplify, because every time we will not perform this particular experiment and we will isolate this particular gene of interest. So better one time when we are isolating, we will amplify this gene of interest. Now, what is amplification? Amplification is nothing but making the similar copies of itself. Now, what itself? We are making the similar copy of gene, that is what called as amplification of gene of interest that we have already digested with the help of restriction enzyme. So suppose you are having this DNA and you are isolating or digesting this DNA with the restriction enzyme. Now what you will get? You will get simply fragments of DNA, because this restriction enzyme had already digested the DNA and we will get smaller fragments. Now you will amplify this fragment because this is your gene of interest. So now once you have amplified this gene of interest, and side by side you are also having vector. Vector is what? Vector is thing that in which you will clone your gene of interest. So the same restriction enzyme you have to use to cleave this vector. So this restriction enzyme will cleave this vector in the same way that is as it has cleaved the enzymes or it has cleaved the DNA. So what will happen? Same ends will be generated. Now this DNA, it will be cloned in this vector. So restriction enzyme ने क्या किया? दोनों को cleave किया. Now this DNA will be cloned in vector. Now this vector is a complete recombinant DNA. So you will insert this recombinant DNA into the host cell. This host cell can be plant, it can be animal, it can be bacteria. Now the last step is obtaining the foreign product. Now once you have made this or constructed this DNA or vector or gene of interest का combination, or you have inserted it in the host cell, surely this host cell will cause the expression of this gene. इसका gene का expression होगा, and you know that in expression of gene you will get the desired protein. So you will get the desired protein out of that gene of interest. So this is what called as foreign product, because this product was not in the cell, but you are introducing it from outside. So at last, we will do the downstream processing. We will purify this product. So now we'll go into detail one by one. So firstly, we'll understand how's the isolation of genetic material, that is DNA or RNA, is done. So here we will focus for DNA. So how this is done? So suppose this is a plant cell, or you can consider it is an animal cell or a fungal cell. So what you have to do, you all know that outside the cell, there is a hard covering or hard envelope that is used to protect the cell. So firstly, you have to cleave this membrane. So because if you will not cleave, how you will get your DNA? And we all know that our DNA, it is present inside the nucleus. So suppose this is a nucleus. So inside the nucleus, DNA will be present. So this is DNA. So we have to take this out. So how we'll do? We'll firstly have to cleave this cell. So we can isolate this DNA either from bacteria, it can be a plant cell, it can be animal cell. So suppose if you are using the bacterial cell, the enzyme that you have to use or you want to use to cleave this envelope is the lysozyme enzyme, because we know that bacteria, they are made up of a cell wall that is made up of peptidoglycan. And how this cell wall is cleaved? It is cleaved with the help of lysozymes. So if you are using the plant cell, you have to use the cellulase enzyme. Why cellulase? Because plant cell, they are made up of cellulose. So plant cell wall, it is made up of cellulose. So the enzyme that will be used to digest this cellulose will be cellulase. And if fungus, so you all know that fungal cell wall, it is made up of chitin. So what you will use? You will destroy this cell wall of fungus, that is chitin, with the help of chitinase enzyme. So how you are breaking this cell wall? With the help of enzyme. So if it is plant cell, you will use cellulose. Fungal cell, you will use chitinase, or as a bacterial cell, you will use lysozyme. So now once this envelope or cell wall is cleaved, now you're having a DNA. So along with DNA, you will also have some other impurities that can be macromolecules like RNA, proteins, it can be lipids, it can be carbohydrate in the form of polysaccharides. So now what is your main interest? Your main interest is DNA. So it's a time you have to purify your DNA or your gene of interest from all other contaminants that is in the form of RNA, polysaccharide, proteins or lipids. So now we'll see how to purify this DNA. So now once we have broken all the cell wall of any plant cell, bacterial cell or any cell of your interest, now it's a time to remove all the RNA, protein and other things that are there in the solution. So how you will suppose you are having this solution in a tube. So how to remove this RNA from here or protein? So what you will add? You will add ribonuclease. What is this ribonuclease? ACE. It is having a suffix called ACE. That means it is an enzyme that will cleave ribonucleic acid or RNA. Why nucleus? Because RNA is a genetic material. So the ribonuclease enzyme will be added to the solution that will cleave all of the RNA that is present in the solution or supernatant. So once this RNA is cleaved, now your DNA ka impurity, it is free from RNA. Now what it has? It again has protein. Now you will add protease that will cleave or enzyme that will cleave the protein part. So similarly, you will cleave other molecules that are present in the solution. And once you are having the pure DNA, now suppose you are having this pure DNA, what you will do? You will add chilled ethanol. Why you are using this chilled ethanol? Because ethanol is an organic solvent that is used to precipitate the DNA. So what all amount of DNA, suppose this is an Eppendorf tube and you are having DNA here, here, here. So what it will do? It will precipitate all the DNA at one place and it will form the clump. And why chilled ethanol is needed? Because we all know that DNA, it is denaturated at high temperature, or the enzymes that is associated with DNA, it has to be maintained at a very low temperature. That is why we are providing it as a chilled ethanol. So it will cause the prevention of all the enzymes that's present in the DNA. So our DNA will be protected from the outside heat. That is why we are using chilled ethanol. So the chilled ethanol is added, it will purify DNA and the DNA will be precipitated out as fine thread. So if you want to isolate DNA, you will get DNA in the form of fine thread. You can see in the diagram, you will get fine thread. बहुत पतला पतला thread you will get. So DNA is in the form of thread, dhaga like structure or in the suspension. So once after adding this chilled ethanol, all the DNA will be collected or they will be aggregated in the suspension as you can see here. So how you will separate out this DNA? You have to separate this DNA by spooling. Now what is spooling? Spooling is a method where you will transfer this DNA or gene of interest by using some kind of synthetic membrane. So what will happen once you will insert this synthetic membrane inside this DNA, it will stick to this synthetic membrane ka part and it will come out. So this is what called as spooling. So you will spool this DNA. So what you have got till now, you have got your you have isolated your DNA, that is your gene of interest. So again, we'll see, firstly, you will do the cell harvesting. That means the cell of interest you will take, whether it is plant cell, animal cell or fungal cell. Then you will do a cell lysis. Whether it is a plant cell, you will use cellulase enzyme. If it is a bacterial cell, you will use lysozyme enzyme. If it is a fungal, you will use chitinase enzyme. That is what called as you are lysing the cell. You are causing the breakdown of the cell. Now is the time for the protein and RNA removal. If it is having the RNA contaminant, you have to use ribonuclease enzyme. For protein, you have to use protease. Now the DNA binding will be there. DNA will be left over in the suspension. You will add chilled ethanol and the DNA will be eluted. This is what called the isolation of DNA. So we have completed the first step. Now, second, we have to make the recombinant DNA. Don't forget, guys, our main aim is to prepare a recombinant DNA. So, how we will make it? Firstly, we have isolated the DNA. So, suppose now I have isolated DNA. So, how I will get my desired gene of interest? Because this DNA segment, it will have many genes, right? So our suppose our interest is this gene part. So what I will do, I will use restriction enzyme that will cleave here only, and I will get this particular gene of interest. So suppose I'm cleaving this or I'm getting this particular gene of interest. So my particular gene of interest is this part. So that's why I'm using restriction digestion. So purified DNA, it is incubated with the restriction enzyme. So now we are moving to the other process, that is what called as next step, cutting of DNA, isolated DNA at a specific location. Who will cut this DNA at a specific location? It will be done with the help of restriction enzyme. And the name itself suggests the enzyme that will cleave the DNA at the specific locations. Welcome, Vinod. Very good evening. So here we are learning about genetic engineering. So I hope you can join from hear from us. So as a result, DNA will digest. How it will digest? Because we have already added the restriction enzyme. So DNA fragments, suppose this DNA is digested with the help of restriction enzyme. And now what we are getting? We are getting fragments. What we are getting? We are getting fragments because this is digested with the help of restriction enzyme digestion. Now, once this fragment that we are getting, we have to select our gene of interest. Now, again, this is a mixed gene fragments. Suppose this is my gene of interest. How I will know this is my gene of interest? Only segregating or resolving these fragments over the gel electrophoresis. Now, what is electrophoresis? Phoresis here means movement or migration. So the migration of charged particle, it can be DNA, it can be protein, it can be RNA under the influence of electric field, that is what called as electrophoresis. Phoresis is movement of any charged particle under electro means electric movement. So what we are doing? We are doing gel electrophoresis. Why gel? Because in the presence of gel, we will see the movement of charged particle. What is the gene of interest here? It is DNA. So we will do the migration of DNA into this electrophoresis. So what gel we are preparing for DNA? We are preparing agarose gel electrophoresis. So the gel, you have a question from here in case of CUET exam. So the gel that we are using for electrophoresis to resolve our DNA fragment is agarose gel electrophoresis. So the gel that we are using for RNA is different, protein is different, but for DNA it is specifically agarose. So this is a star mark for here. So this electrophoresis, it is employed to check the progression of a restriction enzyme digestion. So how to prepare a gel? Suppose this is a casting tray. So what you have to do, this is agarose. This beaker, it contains agarose that is dissolved and boiled in a specific solvent. So once I will pour this gel, after some time it will be casted. It will become solid. So here you can see it is liquid, but after some time it will become solid. Why? Because this gel is semi-solid. So once it is heated up, it will be in the liquid stage. But once I will pour this, or we will pour this gel into the casting tray, this is the casting tray. Once we'll pour this into the casting tray, after some time we'll get a solid like structure. Then where we will load this DNA, because we are saying we will perform this agarose gel electrophoresis. So surely we have to add or load our sample. So where we will add our DNA sample, that will run, because we will run this gel. So where we will add, you can see here comb like structure. This is a comb. Firstly, we have to fix the comb, and then we have to pour this gel. So what it will do, it will create the gel of appropriate sizes where we can load our DNA fragments or our sample. So once this electrophoresis is done, suppose this is gel, this is comb ka well. Now what it will do, after we will provide a power pack or we will supply electric voltage, this DNA will be resolved. So it will resolve like this. Some DNA will be thick fragments, some will be small. So I will make you explain with the help of analogy how this DNA fragments are resolved in the case of agarose gel electrophoresis. So analogy we are taking here. So suppose we have two individual, one is fat and the other is thin. So if I ask both of these individual to run, so who will run? Obviously the thin people or individual, they will run the fast. So similar in the case of DNA gel electrophoresis or agarose, whatever is the smallest or thin fragment, it will resolve or it will move or it will travel more distance than the thick one. As you can see here, the thick ones are retained in the pore size of agarose, but the smaller fragment, they will be resolved one. And once we are knowing what is our gene of fragment, we will take that fragment out and we'll cut this piece of gel from here and we'll resolve this. So now we'll understand more. So we all know that DNA is negatively charged. Yes. So DNA, it is made up of nucleoside, that is DNA, nucleotide bases or nitrogenous bases, that is ATGC, as well as pentose sugar, that is in the form of deoxyribose sugar. And along with it, we also have pentose phosphate backbone. This phosphate backbone, when it is combined with nucleoside, it will make nucleotide. Nucleoside plus phosphate, it will make nucleotide. And what is nucleoside composed of? Nucleoside is composed of sugar as well as nitrogenous bases. What it will make? It will make nucleotide. So this phosphate backbone, it is imparting a negatively charge over the DNA. So where we have to load our DNA. So I've already told this is a gel. So suppose if I'm connecting this to the negative battery. So I will load my sample here because unlike charges, they will attract each other. So if the DNA is negatively charged, I have to connect this terminal, this COM wala well to the negative charge. Why? Because it will move toward the positive charge. It will move towards the opposite charge because the DNA is negatively charged. So it will move toward the anode. And we know that anion is negatively charged, but the anode is positively charged. So again, you will have a question from here if the DNA is negatively charged. If we are loading the DNA towards the negative charge well, so where it will load or where it will move? It will move towards the positive electrode. So why? Because you're applying the electric field or you're performing electrophoresis. So DNA fragments, they are separated according to the size. Again important, how they are separated? They are separated according to the size. As you can see here, the smaller fragments will go down and it will be separated. So they are separated according to the size. So you can see here it is 10,000 and it is 250. According to the size, through the sieving effect of agarose gel. Why sieving effect? Because agarose gel have pores in it. That is why the larger fragment, it is retained here. It is caught in the sieve effect of agarose gel. Again, a star mark question, from where this agarose gel is isolated. So this is a polymer. From where we are getting? We are getting it from a seaweeds or simply microalgae. So this gel, we are getting it from a algae. So I've already mentioned, a smaller size fragment, smaller the fragment, it will move farther. So this process is repeated with the vector DNA. So what all we have done with the gene of interest, we have to repeat it with the vector DNA, because we know that vector is a carrier to carry this gene of interest into the host. So again, we have to digest this vector with the restriction enzyme. We have to run this and we have to resolve this. And once we are getting our defined or desired gene of interest and vector, what we'll do? We'll do a combination of this. And this is what called as rDNA. That is, we are creating recombinant DNA. We are cloning this gene of interest into a vector. So how you will resolve this gel? How you will resolve this DNA fragment? How you are knowing this DNA? This DNA fragments you can't see with your naked eye. So how you can see? So once at the time of preparing gel, you were preparing gel, you have to add one dye, that is what called as ethidium bromide, ETBR. ETBR. That is what called as ethidium bromide. So what is this dye? This dye is carcinogenic. So what it will do? It will bind to the DNA structure and it will be resolved and it will be cleared when you are seeing this DNA under UV trans illuminator or UV light. So how you will see this DNA? So DNA fragment, it can be seen as a bright orange. So the color of this dye is bright orange. So what it will do? It will impart the same color to this DNA fragments. And once you were running this gel, you were not getting this color. But once you will expose this gel under the UV light, you will get the bright orange colored bands. So when they are stained with ethidium bromide and exposed to UV radiation. So again, you may get a question from here. What is the dye that is used to resolve the DNA fragment or used to view or visualize the DNA fragment under UV light? So again, it is ethidium bromide. And again, you can get a question from here. What kind of radiation is used to view the DNA fragment? This is UV radiation. So the DNA, once you find, suppose this is your gene of interest. So what you will do? You will isolate, you will cut this gel from here and you will take it in another tube, because this is your gene of interest. This is what called as elution. So once you will resolve it under UV light, you will cut this gel. So this is what called as illusion. So DNA bends, they are cut from the agarose gel. This is called as illusion. So now, if you have repeated this for vector also, what you have, suppose this is gene of interest, you have cut it with the help of restriction digestion. This is your vector, and again restriction digestion is done. Now you have your recombinant DNA. So how you will fix this ecoriga or any gene of interest with the vector? It is with the help of ligase enzyme. How you will join this gene of interest and vector? It is with the help of ligase. Means enzyme, that is ACE, that is used to like something. Like means to join, that is used to join. And this ligase is joining what? This is joining both gene of interest after restriction digestion as well as your vector, that is a carrier. Now it will create your recombinant DNA. So we have already seen isolation of DNA and how this recombinant DNA formation is taking place. This is taking place with the help of restriction digestion. Now you have your gene of interest. What you have to do? You have to amplify your gene of interest, because anytime you may need this gene of interest. So the third process is amplification, that is making the copy of it. So how you will do? You will do it with the help of polymerase chain reaction. Now what is this polymerase chain reaction? This is a technique which is used to make the copies of DNA, where you can make multiple, as many as number of copies of DNA according to your interest. So here we have to use the primer. I will come to this point and how it is used to amplify this DNA. We will amplify this DNA with the help of enzyme called as DNA polymerase. So what are these primers? So these primers we are synthesizing it in a lab. That is why it is called as chemically synthesized oligonucleotide, that is complementary and opposite to the regions of DNA. So now we'll understand what is amplification. In amplification, we know we will perform PCR, that is polymerase chain reaction. That means a chain of reaction will be performed that will polymerize our gene of interest. That is why it is called as polymerase chain reaction. And under this technique, we have three steps. First is denaturation, again very important. Second is annealing, and the third one is extension. First, you have to denature the DNA. Annealing of primers is done, and the extension. Now we'll see the process one by one, how this amplification is being done in case of PCR. So the very first step that I have put here is denaturation, and exactly in PCR you will have the very first step, that is denaturation. So suppose you have this double-stranded DNA or your gene of interest. So what I will do, I will firstly do the denaturation process. Denaturation, that means something that is nature, you will remove that. D means removal. You will remove that organization. So what you are doing, you will remove these or separate these two strands of DNA. That is what called as denaturation. How you will do is exactly, you will heat the target DNA or gene of interest at very high temperature. Again, an important point, what is a higher temperature to melt this or to separate this DNA strand? It is 94 degrees centigrade to separate the strand. And once these strands are separated, now both the strands will be used as a template for the process of DNA synthesis. So the very first process is denaturation. How? Because you are heating the target DNA at 94 degrees centigrade. Now the second process is annealing. What you have to do in annealing? Annealing means to join something, to anneal something. What you will anneal? You will anneal primers. From where these primers are getting? We are getting these primers, we are synthesizing it in the lab. That will be complementary or opposite to this template DNA strand. So what I will do? What I will do, suppose I have two strands of DNA, I will take two sets of primer. That is why it was mentioned two sets of primer. Two sets of primer. One will be the forward and one will be the reverse primer. So we will use two set of primer here. So this is one set and this is second set. Where it will be annealed, it will be annealed always to the 3' end of the DNA template. So here we are having 3' end and here also 3' end. Why 3' end? Because we have to maintain the complementarity. That means the opposite side. Always we have to synthesize the DNA in the 5' to 3' direction. So how we will do? We will do once we will anneal this primer in the right orientation. That means it in the 5 dash to 3 dash. So here you can see 5 dash to 3 dash. How it will be done? Jab opposite complementarity hoga 5 dash and here 3 dash. That means it is opposite. 5 dash of primer, 3 dash of DNA. So it is the joining of two primers, two sets of primer to two different DNA strand at the 3 dash end just has to maintain the complementarity. Again, very important point, you may have a numerical for from here. You have to calculate the annealing temperature, and in the question you have given the melting temperature. So how you can calculate the annealing temperature? So to calculate the annealing temperature, the formula is melting temperature minus 5. So when you will minus 5 with the value that is given in the question, this will give you the annealing temperature. So the annealing temperature formula is melting temperature that will be given in the question minus 5. And vice versa, if you have to calculate it for melting temperature, so annealing temperature plus 5 will give you melting temperature this time. So the third process is extension. So we have seen denaturation, annealing, and the third is extension. Now, how this extension is done? Extension means you are extending this primer and you are making full complete copy of DNA. So who is doing this? DNA polymerase. So why we have given this RNA primer then? If DNA polymerase is doing this step, because RNA primer, DNA polymerase is an enzyme which cannot synthesize DNA on its own. It needs the help of a primer. So that's why we are adding primer, and using this primer, DNA polymerase will synthesize or polymerize this DNA. And the DNA polymerase that we are using here is a hot spring DNA polymerase, that is Thermus aquaticus. Why hot spring? Why not we are using simple DNA polymerase that we are having inside? Because this DNA polymerase has to bear 94 degrees centigrade temperature that we are providing during the process of denaturation. Because if we will not provide this, how the denaturation of strands will be taking place? So who will bear this much temperature? So it will be bared by those polymerases that are isolated from hot spring. So the name itself is Thermus aquaticus. Hot spring matlab water. Aquaticus means water that is isolated from hot water or hot spring. That is what called as Thermus aquaticus. Again, this is an important question. So what it will do? What is the significance? It will remain active in high temperature during the denaturation of double-stranded DNA, because this is an enzyme. So what it will do? It will cause the DNA polymerase that is isolated from Thermus aquaticus. This enzyme, it will get denatured in higher temperature. But this is isolated from higher temperature itself, so it will resist this 94 degrees centigrade temperature during denaturation. So once this DNA polymerase chain reaction is done, at the end we will get 1 billion copies if we are running it for 30 cycles. So here I've explained it for one cycle: denaturation, annealing, and extension. So once this annealing is done, DNA polymerase from Thermus aquaticus, it will anneal or it will extend this DNA. So after 30 cycles, we'll get up to 1 billion copies of DNA. So can you imagine, after isolating this small fragment, you will be getting a 1 billion copies of DNA. Now you will clone this DNA into a vector, or you can also keep this gene of interest for your further use. Now you have your recombinant DNA. Now what you will do once this amplification is also done, it's a time to transform this recombinant DNA into the host cell. Now this time you can use this host cell, can be a bacterial. Most of the time we are using E. coli. It can be insect cell or a plant cell. So how we will do this process? So suppose I will zoom this process. So I hope you can see this. So suppose if I am transforming this gene of interest, this is a vector and this is gene. So this is gene. Red one you can see here, it is a gene. So this is what called as recombinant vector. We have inserted this red wala part in the green side, that is what called as gene. Now here we will have two possibilities. Either we will get a non-transformant or transformant. Under non-transformant, we will not get this whole vector or recombinant vector into the host cell. So suppose this is a host cell. This host cell is having this black one own DNA. But we are not getting this DNA. You can see this diagram is not here in host. This is what called as non-transformant. But if we are getting this DNA here, this is what called as transformant. So I hope this is clear to everyone. Non-transformant, nothing we are getting, and transformant, at least something we are getting. And under transformant, again we have two possibility. We can either have non-recombinant or recombinant, because every time we are not sure that our gene of interest is entering in the vector. If our gene of interest is not entering in the vector, this vector, that is what called as non-recombinant. As you can see in this diagram, this red part is not here in this green part. That means only vector has come, but no gene of interest is incorporated in the vector. That is what called as non-recombinant students. And then what is recombinant? Now this recombinant one is the desired one, where this structure is coming, and along with this vector you can see a red mark in this green area. That is what called as gene of interest. So here what we are getting, this we are getting recombinant. Now we will select this recombinant colonies from here. Now, once we have got our recombinant DNA or expression of recombinant DNA in the host cell, it can be animal, plant, or E. coli, that means bacteria. Now, it's time to get the foreign product. Now, how product? Now, suppose we are having this gene, it is incorporated into the DNA of host cell. Now, once this host cell will replicate, it will make its own DNA or it will make RNA and protein. That time we will also have the protein of our gene of interest, because we have incorporated this gene of interest into the host cell. This is what called as foreign product, because this product, it was not present in the body of your, in the host of whatever host we are using, whether it is bacteria, animal, or plant. So the ultimate aim of recombinant DNA technology, now we have made a recombinant vector. So the ultimate aim is to get the desirable protein. From where we will get this protein? We will get this protein from a recombinant DNA or recombinant vector that we have already made. So here we will use a heterologous host. Why host? Because we are using some other host where we are incorporating or introducing this recombinant DNA. So this host is called heterologous. Heterologous meaning because this gene, it is not present in a host, but we are introducing it from outside. That is why it is called as hetero, means different host, because this host, this DNA, it was not present in the host. Now it will make the recombinant protein. So whenever this host will express its DNA, it will make this DNA also or protein also. So this is target gene. Now we have made the plasmid or vector. We have incorporated this gene of interest in a vector. Now this is called as recombinant DNA. And once we will use it in a host, suppose this host can be mammalian cell, insect cell, it can be yeast, or it can be prokaryote. So the most of the time we are using E. coli, bacteria, or prokaryote. Now what we will get? We will get the recombinant protein. So the recombinant protein that we are getting, it is in a small amount. So suppose if I want a larger production of this enzyme, what I will do? I will put it in a culture vessel. So what is this culture vessel called? This is called as bioreactor. Bio means something living organism. Reactor means that will cause the mass production of this protein. So now we will understand how we will do the mass production of this protein. So the cultures are used to extract desired protein. This we will use culture in this vessel or bioreactor. We will use some culture that will give the desired protein ka extraction or more amount of protein, and then we will purify it with the help of separation technique. So what we will use? We will use bioreactor here. What kind of bioreactor? It can be a continuous culture system. Continuous means continuously we will provide a medium from here, and the spent medium that is used by the microorganism or protein, we will remove it from here. All time we are providing extra of fresh medium, so that our cells will be there in the fresh medium and we will get a larger amount of product. So what we will get? It will produce a larger biomass, it will yield more desired protein. So what we'll use? We will use bioreactor. As you can see in the diagram, this is an example of bioreactor. So you can see this is kind of a machine. So these are vessels in which raw material are biologically converted into specific product. So what you will do? You will mix your microorganism, you will mix your specific gene of product. They will convert this product into a more amount. So what you will do? You can create your 100 to 1000 liters of product. What you have to do? You have to do this culturing. You have to put this much culture into the bioreactor, and you can just imagine, if you are using this much culture, you can get larger amount of product here. So but make sure you have to maintain an optimal growth condition. You have to maintain temperature, pH, because it is very important to maintain the temperature for the cell, pH for the cell, because if the pH is altered, cell will not grow. Substrate, salts, all kinds of vitamins, and oxygen. Make sure, because our cell is aerobic, it is always respiring, so make sure you are providing the oxygen from outside environment. So there is one port from where you can also provide this oxygen. So the most common type for this bioreactor is stirred tank reactor. So the most common type, what it will have stirrer. This is a stirrer. What it will do a continuous mixing of this culture. What will happen? This, if this culture is continuous mix, so this small dot, it is cell. So once this culture is mixed, this culture or media will be sent to all of the cells, and the cells, they will produce the greater yield of product. But if you're not doing this mixing, or you're not using this stirrer type bioreactor, what it will do, this continuous mixing will not be happen. So if it is not happening, so we will not get that desired product in large amount at the end. So it is usually cylindrical. This vessel is cylindrical, or a curved base that will facilitate the mixing of the reactor contents. So this stirrer, you can see stick like here vertical. This, it will facilitate even mixing, it will do proper or uniform mixing and oxygen availability. Okay. So what are the parts of bioreactor? Agitator or stirrer that will mix everything. Oxygen delivery system, so as to provide the oxygen. A foam control system, you have to prevent the foam formation. So what will happen once this medium is mixed with the help of stirrer? What it will do? It will cause foam generation or leather formation. So as to prevent this formation, so what will happen? If this foam formation will happen now, all the cells or the proteins will not be functional. They will break off. So to prevent this foam formation, we have to provide the foam control system so as to prevent the foam formation during this mixing up process. Also, the temperature control system is needed. We have to maintain the temperature if it is going high, pH control, and also sampling ports where you will add your fresh medium and you will remove your spent medium that is used by the cells or the protein. So once you have got your all your proteins in a bioreactor, then it's time to process or purify your protein. So that is what called as downstream processing. So we have two kinds of processing. One is called upstream and the other one is called downstream. Upstream, all the procedures or step that you are doing before this protein formation is what called as upstream. So what you are doing here, you are isolating your DNA, point number one. You are cutting your DNA with the help of restrictionization, that is two. You're also cutting your vector, you're preparing your vector, and ultimately you're preparing your recombinant DNA, and you are amplifying your gene of interest. So all these process will be called as upstream process. But when you are doing this protein formation step, so what it will be called? It will be called as protein formation or desired product formation. Now purifying this product, it is what called as downstream. Down means afterwards. Afterwards, our ultimate aim is protein. Listen guys, our ultimate aim is product. What product? Protein. From where we have got this protein? Gene of interest. So what all things we will do to purify or separate this protein from other contaminants or impurities, that is what called as downstream processing. So what we'll do, we'll do cell lysis, whatever cell microbial cells will be there. So suppose if I've incorporated my recombinant DNA into the E. coli. Okay, so suppose this is my gene of interest and this rectangle part is, sorry for the annotations guys, so I hope you can understand everything. So suppose this rectangular part is E. coli or a bacteria, and we have this recombinant vector here. What I will do, I will put this many bacteria. I will culture this bacteria in the lab, and many bacteria will be there, this rectangle part. I will put it in the bioreactor, and what I will get? I will get many bacteria that are replicated or formed. And inside this, I will also have my product, because you can see inside this, the DNA is there, so it will form the product. So what I will do at last, suppose I'm getting this massive production in the bioreactor. What I will do, I will harvest all the E. coli red dot from here, correlate this red dot with the help of this rectangular part. So this is E. coli. So I will isolate all the E. coli and I will break this cell. This E. coli cell, I will lyse it, I will break it off, and the what all products will be there, I will purify it. So this is my purified products. So here I will get the purified. So what is downstream processing? It is a series of processes such as where we are separating and we are purifying our products after the biosynthetic phase. What is biosynthetic? The phase from where we are getting the protein. So this is the actual biosynthetic phase. And what all processes we are doing after this yellow part, a protein formation step, or in a bioreactor, that is what called as downstream processing. So now I hope and or I'm sure this topic is clear to everyone. So what all we have studied? We have five processes under genetic engineering. Firstly, you have to isolate the DNA, and once your DNA, pure DNA is isolated, now it's a time for the restriction digestion of both the gene of interest or the carrier that you are using to incorporate this gene of interest in the host. So the second is restriction digestion. Now after this restriction digestion, we will amplify this gene of interest. How we will amplify it? We will amplify it with the help of PCR. Now, inside the host, inside the living system, we are having the replica machinery. But if we have to isolate this DNA and multiply or amplify this DNA in the lab, how we'll do it? We'll do it with the help of a machine or the artificial machinery, that is what called as polymerase chain reaction. Will replicate the exact phenomena as like it is happening it in the living cell. And the fourth process is, now, once this amplification is done, now the fourth process is recombinant DNA ka formation. Once this recombinant DNA formation is done, we will incorporate this recombinant DNA into the host cell. This host cell can be E. coli or bacteria. Suppose this is E. coli. What we'll do, we'll put this E. coli into the culture medium where E. coli will go. At the same time, our desired protein will also, a product will also grow. So we'll now do downstream processing, that is what called as separation and purification of the product after the biosynthetic phase. Now we'll practice two or three questions so as to get an idea from where you will get an question in NEET examination and what are the type or pattern of question that is asked in CUET UG examination. So if you have any doubt, you may post your queries here. So the question number first is the most common type of gel that you can use for DNA separation. As I've already mentioned during the time when I was explaining, the most common type of gel that you can use it for DNA separation is it agar, is it agarose, is it beige, or is it all of the above? So what is the most common type of agarose gel for the preparation of or for the separation of DNA fragments? Very good, Sneha. This is agarose. Because here we have not discussed about beige, so omitting this option itself will omit 4 because it is saying all of the above. Again, agar is not there. What we are having, we have studied about agarose. Agarose gel electrophoresis, that is the main gel that is used to separate the DNA fragments. So now we'll move to the next question. What is the current sequence of a PCR reaction or amplification of DNA? So I've explained you three process of PCR: first, second, third. And here you have to select the correct sequence of method in which the PCR reaction is being followed. That is polymerase chain reaction. A. Yes, Sneha, you are correct. The correct sequence is denaturation, which will cause the separation of DNA fragments. And next, we will anneal our primers and then we will synthesize the DNA with the help of DNA polymerase enzyme. So, the very first process is denaturation. So, you cannot find denaturation in any other option. So, all three options goes wrong and the right answer is the option first. So, which dye is used to visualize DNA using electrophoresis? So, very simple question. What dye can you use to visualize your isolated DNA fragments? Because you cannot see your fragments under DNA gel electrophoresis. So, you have to visualize it and you have to isolate or elute your gene of interest. So, how you will do it? Is it Kumasi brilliant blue? No, we have not at all studied. Is it bromophenol? No. Methylene blue? No, we have not at all discussed. Very good, Sneha. It is styrene bromide. This is carcinogenic to human. This is a orange color dye. And once we'll add it to the DNA, it will bind to the DNA and after the gel run will expose this DNA under the UV trans illuminator. What it will show? It will show all the DNA fragments in an orange color. Why? Because this dye will bind to the DNA. So the right answer here is fourth, that is ethidium bromide, that is orange in color. So then the last question that you have is you have to calculate the annealing temperature. So what is the question here? What would be the preferred annealing temperature for a polymerase chain reaction? Be careful. Here you have to calculate the annealing temperature. If the melting temperature for the oligonucleotide is 63.1. So I've already mentioned you what should be the formula to calculate this annealing temperature. So all of you guys, you should please try all this question. This is very easy if you can make it down because I've already mentioned the formula for calculating the annealing temperature. So what is the formula? What is the formula for the annealing temperature? Okay, Sneha, let's see. So what is the formula? That is annealing temperature, that is T is small a, annealing temperature. It is Tm minus 5 degrees centigrade. And what is Tm? This is melting temperature or denaturating temperature. So Tm here, it is given melting temperature, that means Tm. It is given 63.1, but we will separate if this from 5 because Ta is always Tm minus 5. If we will subtract it, we will get 58 degree. So near about 58.1 or 57.9 something you will get. So this option goes right. That is option A. So I'm sure this topic is clear to everyone. So we have also practiced numerical, like what is the gel that is used for DNA separation, what is the reaction or steps for PCR, what is the dye that is used for DNA electrophoresis, and how to calculate the annealing temperature. But if it is asked in the other way, if the annealing temperature is given and you have to calculate the melting temperature, this would be annealing temperature plus 5. That will give you melting temperature. So if you have any query, you may ask here. Thank you, Sneha. So, okay. So, thank you everyone for watching this session. If you like the session, you may like, share, and subscribe. You can also share this link to all your friends whosoever is preparing for this CUET UG exam. Or if you have any query, you may post your query in the comments section, or you can also mail to us anytime. So, thank you once again for your time and patience. Thank you once again. Wish you all the best. Bye bye. Take care.